Okay, so we want to try to block polyspermy. There's going to be two ways of doing it. What we call a fast block, which happens in a few seconds, and then a much slower but more thorough, what we call slow block. The mechanisms are completely different. What triggers them off is completely different. But let's take a look at the fast block first. Okay, what's going to happen is this. Aside from activating that sodium hydrogen co-transport, we're also going to open up sodium channels in the plasma membrane, so still more sodium rushes in. Now in this case, we're just allowing facilitate diffusion of sodium, a positively charged ion, and this time we're not kicking anything positively charged out. The co-transport system, one sodium goes in, one H plus goes out. So overall, the charge across the membrane stays the same with that co-transport system. But here we're just letting positively charged ions in, and we're not taking any more positive ions out. So what's going to happen here? We have okay, cells, and you remember this from cell biology, cells universally, this includes prokaryotes as well, are electrically negative relative to the outside. In other words, they have fewer positive ions overall than the outside does. And generally that membrane voltage is on the order of about 50 millivolts negative, about 20th of a volt. Now it doesn't sound like much, but when you're talking about that kind of electrical potential across something, the thickness of a cell membrane, which is only 5 nanometers thick, the electric field strength is a couple 100,000 volts per centimeter. That's tremendous. In case in point, air breaks down at 25,000 volts per centimeter. That's why you get a static shock. You know, that nasty static shock you get in the winter. That can have more voltage than the electric chair that Alabama used to use. Fortunately, amperage is a lot less than that. But, you know, it can be painful, trust me. I've given myself some real jumps from good static shock. But anyway, what you're doing, you have that electrical charge across a very thin electrical insulator, which of course is a very hydrophobic cell membrane. So ordinarily, you're hovering around, say, 50 millivolts negative. And now, all of a sudden, sodium rushes into the cell. The membrane voltage in the A goes up to maybe about plus 10 millivolts. So instead of being negative, now it's slightly positive. Now those of you who remember nerve cell physiology, you see the same thing except even more dramatic. Okay, now what happens at that is some of the proteins in the membrane are going to change their tertiary structure when the voltage across the membrane changes. I mean, if you're talking about that kind of electric field strength, think of what that's going to do. All of a sudden, switching these charges from, say, you know, the equivalent of a couple 100,000 volts per centimeter, and then switching it to practically zero or maybe a little bit above or whatever, that can distort, that can pull and push away charged amino acids and things like that. That can affect protein tertiary structure. In cell biology and physiology, those voltage-gated ion channels, the sodium and potassium ones that are involved in nerve transmission, that's an example. They open and close depending on what the membrane voltage is, and that triggers off nerve transmission down the axon. Okay, so this is another example. Well, what happens when you do that? Well, it turns out the sperm receptors bind quite well when they're in a membrane that's electrically negative. On the other hand, change that voltage and bring it up to slightly positive, it distorts the sperm receptors and they can no longer bind sperm. Okay. Now there's an interesting little experiment that kind of shows this. There is an electronic device called a voltage clamp. Let me just put that over here. It's widely used in neurophysiology. People that study nerve cells and nerve physiology often use this. What a voltage clamp is a little instrument that has two tiny little electrodes. One of them you stick into the cell of interest, the other you stick on the outside of the cell, touching the membrane of the cell. And then you have this dial, and you can adjust, it will send an electric current between those electrodes, and you can adjust the voltage across the membrane to whatever value you want, at least within reason. Too much, you'll tear the membrane apart. But anyway, what it does is allow you to set the membrane voltage. And once again, neurophysiologists use these guys a lot. Okay, well what somebody did is, hey, well we noticed this voltage change and that seems to prevent sperm binding. Let's test this out. So what they did with the voltage clamp is first, they held the voltage at -50, added sperm, and lo and behold, when they did that, they got lots of polyspermy. Now, on the other hand, if pre-fertilization, they voltage clamped this guy at, say, 0 or 10 millivolts, then they added sperm, they found fertilization almost never occurred. So that told us something very important. That little experiment told us something very important. The voltage change across the membrane alters the sperm receptors in such a way that they can no longer bind sperm. If you keep the voltage negative, voltage clamp, or you prevent that sodium influx, you're going to get polyspermy. If you keep it high, you won't get fertilization. At least you won't get much of it. So, that is our fast block to polyspermy. That change in voltage which distorts the structure of the receptors, and now the sperm don't recognize those receptors. Okay, and that happens, like I say, in a few seconds. Now we have the slow block. This is the one that really is going to prevent any subsequent fertilization. And this is what we often call the cortical reaction. Okay, the calcium ion level in the cytoplasm skyrockets from micromolar to millimolar, because calcium is rushing into the cell, and calcium is also being released from internal stores. Now, a kind of cool thing, I've seen videos of this, this looks really, really neat. It turns out there is this particular dye that ordinarily is colorless, but it fluoresces, it glows in the dark, kind of this pretty dark blue color, this glowing blue color, in the presence of calcium ions. So you take this dye and put it in a buffer and look at it in the dark, under black light, ultraviolet light, you see nothing. But add calcium and it just lights up this beautiful blue-white color. Now, what you do is you inject this into an egg, like a sea urchin egg, which is semi-transparent. You can do it with frog eggs too, but it's better, looks cooler as sea urchin eggs. Okay, so now you got this stuff, it's loaded up with dye. And not surprisingly, because the levels of calcium in cytoplasm are so low, the stuff doesn't glow. And now you add splane, and in comes the sperm. And it hits the sperm, the first sperm that gets in. You start suddenly seeing this blue glow coming right around the sperm entry site where we're getting the calcium release and the calcium influx first. And then it just, in a matter of seconds to a couple minutes, it propagates completely around the egg cell. And it just looks so cool. You see that little blue flash, and then it just expands around the entire egg cell. And what it's telling you is we're getting this propagating release of calcium ions. Because it turns out those internal receptors, the ones that bind IP3, they're also triggered off by calcium ions on the outside. So you release calcium ions, and that causes still more of those receptors to open. It just propagates like a wave. I don't know if you've ever seen, sometimes you see in these science fiction films, the nasty aliens come and they fire their phasers or photon torpedoes at a planet, and then you just see this flash and the whole planet starts lighting up and just ignites in a ball of flame, propagates across the whole thing, and then boom, and bye-bye Earth or whatever. Okay, cool special effects. But when you see this, that's what it looks like. That's what it looks like. It's so neat to actually see this kind of thing. So we get that propagation of calcium release. Now, calcium does all kinds of things in cells, but the first thing we're going to look at is the so-called slow block to polyspermy. Once again, we have those calcium-regulated proteins that connect from the membrane of the cortical vesicles to the plasma membrane. In the presence of calcium, they contract and kind of coil up like a spring that pulls the vesicle close to the plasma membrane. Other proteins grab on those membranes and they fuse together, and now simultaneously all these vesicles secrete. Okay, once again, calcium ions are critical for all kinds of processes where the cells secrete things. You need them. So now, this is what we call the cortical reaction. So what I'm going to do is I'm going to play around with these membranes a little bit. And this, of course, represents these cortical vesicles fusing and releasing their contents. So the contents are being released out. And this, of course, is what we call the cortical reaction. All right. What does the cortical reaction do? What is in those cortical vesicles? Okay, let's look at the ingredient list in cortical vesicles. First of all, it's got protein-digesting enzymes, or proteases. As a matter of fact, they're in the family known as the serine proteases, which many proteases fall into that family. They're called serine proteases because all of them have at their active site the amino acid serine just in the right location, and that is actually what catalyzes this reaction that cuts one amino acid from the next. And if you screw up that serine in that active site, there's actually a protease inhibitor that does just that. It covalently attacks the serine, and it kills that protease dead, dead, dead. I mean, it's irreversible. It kills the protease, it can never come back. It can trash the active site. Okay, well, these serine proteases. Now, trypsin is a classic example of a serine protease. That's why we could do that egg experiment. We could add trypsin inhibitor, and that inhibits these proteases as well. And if I were to add that stuff called PMSF, that would do the same thing, except PMSF is toxic as all get out. And it's very insoluble in water. Well, it would do the same thing because it would destroy the active site of all serine proteases. Okay, so proteases, what do these guys do? These guys, the ones in the cortical vesicles, are not just general proteases. They're fairly specific. You know what the target is? Sperm receptors. They don't just chew up every protein in sight. They specifically attack and destroy the sperm receptors. Okay, now the second thing we've got sets of enzymes that are basically going to chemically join glycoproteins together. And in a sense, almost like what formaldehyde does when you add it, to embalm something or preserve a specimen or whatever. Formaldehyde chemically cross-links various biological molecules together. Okay, so these enzymes cross-link glycoproteins. And what that does is it hardens the vitelline membrane. Oops, especially if you can spell it right. Additionally, these proteases not only do they trash the receptors for the sperm, the proteins that connect the plasma membrane to the vitellin membrane are also attacked. So we'll say connections to vitellin membrane. And then, last but not least, the cortical vesicles also have lots of polysaccharides in kind of what you call almost like a dry or powder-like form. In fact, many of these polysaccharides are sulfated. Now recall, polysaccharides are extremely polar molecules. Sulfated ones even more so. That's why we use them a lot in extracellular matrix. So what happens if I were to take some dried polysaccharide and I throw it into water? The stuff's too big to actually dissolve in water. What it does is it tracks water and soaks it up like a sponge. So for instance, we do that gel filtration experiment in cell biology. If I take 10 grams of that, what they call Cefidex, which is a polysaccharide, if I take 10 grams of it and throw it in half a liter of water, I will turn the stuff into kind of a semi-jelly-like thing. Okay, so these things soak up water like a sponge. So what's going to happen here? We've got this stuff that soaks up water, it swells up. We've got something that trashes the sperm receptors and the connection between the vitellin membrane and the plasma membrane. And then we got things that cross-link the vitellin membrane glycoproteins and polysaccharides together. So, what's going to happen here? All right, let's take a look. I'm going to get rid of the follicle cells here. We don't need them right now. Okay, here's our vitellin membrane. Okay, we've got these connectors. We've got these sperm receptors. Okay, out go the proteases. Bye-bye connectors, bye-bye receptors. So we get rid of all of these guys. Now the vitellin membrane is no longer physically connected to the plasma membrane. Meanwhile, our cross-linking enzymes are going to turn this somewhat formerly fragile membrane into a layer of armor that's very hard and resistant to physical stress, and it's also resistant to most forms of proteases. And then all those polysaccharides that go in here absorb water. Don't forget, these membranes are very, they're totally porous to water. That vitellin membrane, water can go right through it like it isn't even there. Okay, so what happens now? We've hardened the vitellin membrane, we've cut all the connections, we've trashed the receptors, and now those polysaccharides swell up. And they push that former vitellin membrane out. And now it becomes easily visible under the microscope, and we call this a fertilization membrane. It's the former vitellin membrane, but now it's on steroids. It's been cross-linked, so it's tough and durable. It's been pushed a significant distance away, and there's no receptors associated with it anymore. We trashed those receptors. Okay, now what happens? This is like tank armor, if you happen to be the perspective of the sperm. What happens? The little spermie comes swimming along. And it comes swimming along, and it, boom, bumps into that fertilization membrane. It's not going to fire off its acrosome because there's no receptors there. And even if a few are left over, it fires off the acrosome. It's like shooting a Glock at a tank. It ain't going to work. It ain't going to penetrate. So you can have all the acrosomal enzymes in the world, and they ain't going to cut through that anymore. So now we have a complete block to polyspermy. Okay, that's the cortical reaction. That's our slow block to polyspermy. Now how slow? You look at echinoderms, it's about 30 seconds. It starts in about 30 seconds and finishes in another 30 or so seconds. That's why once in a great while, somebody in the class actually gets to see one of these as it occurs. But you have to be real lucky. Now, unfortunately, with the sea urchin eggs we use, this other species, though they're much more fertile than the ones we used to get from Carolina. The problem is when that fertilization membrane only goes out about 10% of the egg diameter. The nice thing about the other ones, on the rare occasions you could actually get them to shed gametes at all, was the fertilization membrane went out fully 1/3 of the diameter of the egg. It was just, if you couldn't see it, you need to go to an optometrist. Okay, this one's a little hard. That's the one drawback with these guys. But on the other hand, you know, you don't have to go through a dozen sea urchins and not get anything fertile. Yes?
Okay, so the telemembrane swells because all the contents from the vesicles just like fills it up.
Absorb water and swell up. Yes, so expand. They expand a lot of volume. They come in dry, suck up water like a sponge, including, you know, water is going to come in through the vitelline membrane from the outside because it's so porous. So it just swells up. We don't have any more connections attaching to the plasma membrane. So it swells up and kind of stretches a little bit and just expands. Okay. All right. Now, the chynoderm's done about 30 seconds. If you look at things like frog eggs, it usually starts about 2 minutes and takes a couple more minutes to finish. If you look at mammalian eggs, it tends to be more closer to about 5 minutes. Now, by the way, for you medical types, the cortical reaction in mammals is called the zona reaction. But it's essentially the same kind of thing. And that is our complete block, this polyspermy. In very high sperm concentrations, that fast block is not totally foolproof. It certainly cuts the chances of polyspermy down a great deal, but it's not an absolute barrier. This is pretty close to an absolute barrier. As long as that fertilization membrane is physically intact, sperm ain't going to get into that stuff. Zona reaction. Zona pellucida equals vitelline membrane. Okay, yeah. Who wants to go to med school, right? Yeah. Okay, all right. Here's zona reaction. It's a cortical reaction with a bunch of fancy Latinese so that only physicians can understand it. That's why they do that. And then they get charged like $150 a visit. Okay. All right. Okay, so these are our polyspermy blocks. And once again, the events that cause this are twofold. The calcium influx and calcium release from the inside, that causes the cortical reaction, or the zona reaction. The sodium influx that happens within a second or two of the receptors, the egg sperm receptors coming in contact with each other, that happens within a second or two, and that gives you the fast block to polyspermy. So we've got these two blocks to polyspermy. Now, there actually even is a third backup system. This is when all else fails. What'll happen, let's suppose by chance a couple guys slip through the crack. Okay, so now we got two sperm trying to fertilize the same egg. The first one that gets in is somehow going to trigger off some kind of, I don't think we really know the mechanism of this very well, but triggers off some kind of thing that the second sperm nucleus is, or subsequent ones, are destroyed so they never have a chance to fuse with the egg nucleus. Now here again, once in a great while, even that can be overcome. But we have a three-layer defense here, which is a good thing considering what happens developmentally if you get polyspermy. In other words, think of this. You know how bad a trisomy is, where you have 3 copies of chromosome 21 instead of the normal 2. That causes all kinds of developmental issues leading to mental problems, premature aging, cardiac problems, and stuff like that. Most other trisomies, we're talking a single chromosome here, are embryonic lethal. You don't even get to be born. Actually, a lot of trisomy 21s result in miscarriages too. But anyway, okay, so that's one person. Imagine having an entire set of all chromosomes, trisomies of everything. You can imagine what that's going to do to development. It's really going to screw everything up fast and thoroughly. So that means developmental arrest generally an extremely early stage of development. So we want to prevent that. So that's why we do a multi-level defense here. If one don't stop it, the other will stop it. If those two don't stop it, we have even a third emergency backup mechanism. So we have a plan A, a plan B, and a plan C to prevent the tragedy of polyspermy. Okay, well, we've got that. Now other things are going on. Finally, hopefully that one sperm goes in and binds to the plasma membrane receptors. Okay, cortical reaction is over with. Here's the spline. And here's some receptors. And here's the second set of receptors. They interact with each other. That's going to start triggering off changes in the cytoskeleton around here as well. Now, I'm not quite sure the signaling pathway. There are many different signaling pathways that can actually alter the cytoskeleton. But what's going to happen here is the sperm is now attached to the egg plasma membrane. Now we have to take that sperm in. We've docked, now we've got to get it into port. We've got to tow it into port here. Okay, now what's going to happen is we're going to see this is the actin microfilament part of the cytoskeleton. Actually, by the way, I should probably...
Oh, Lord, yes.